What is recombinant DNA technology? Recombinant DNA technology is the joining together of DNA molecules from the two different species. The recombined DNA molecule is inserted into a host organism to produce a new genetic combinations that are valued to science, medicine, agriculture, and also industry. Since the focus of all genetics is the gene, the fundamental goal of laboratory geneticists is to isolate, characterize, and manipulate genes. Recombinant DNA technology is based primarily on two other technologies, which is cloning and DNA sequencing.
So how recombinant DNA is useful? Through this technique, bacteria has been created which capable of synthesizing human insulin, growth hormone, alpha interferon, and many other medically useful substances. Other than that, recombinant DNA technology also can be used for gene therapy, in which a normal gene is introduced into an individual's genome in order to repair a mutation that causes the genetic disease.
Next, I'll be explaining about DNA cloning. A clone is a group of individual cells or organisms that came from one progenitor. The word clone has extended to DNA recombinant technology and has provided scientists the ability to produce multiple copies of a single fragment of DNA, such as genes, creating identical copies that constitutes a DNA clone. A small replicating molecule is called DNA vector. The most commonly used vectors are plasmid, viruses, and yeast cells.
For DNA cloning, first we want the gene that we will clone from a double-stranded DNA. Then we need to have restriction enzyme to cut the gene out. The restriction enzyme will latch onto here and identifies the genetic sequence and cuts in the right places. And we use another restriction enzyme that identifies with the sequence at the other side. After applying the restriction enzyme, we retrieve only that part of the gene. We then paste it onto a plasmid. The gene will have overhangs that allow the plasmid that have complementary base pairs over the overhangs that will make the plasmid react easier with each other. DNA ligase will also be put in to connect the backbones over here. Now we have this plasmid that we want to insert into an organism that help make the copies for us. We typically use E. coli. Now we put the plasmid inside a solution that has E. coli in it. We will then apply heat shock for the bacteria to take up the plasmid. The bacteria that has taken on the plasmid will be transferred to a plate that has nutrient. This allows the bacteria to grow into colonies. The colonies of bacteria will have ones that did not take the plasmid, so we need to isolate the bacteria that has the clone DNA. There are a few ways to do the isolation. One of the ways is to put gene that has antibiotic resistance inside the plasmid. So, instead of having only nutrients inside the plate, we will also be putting antibiotic. Thus, we have colonies that we target that are susceptible to the antibiotics, and the bacteria that did not take the plasmid will not grow. Now you can take the colony and continue to grow it to have multiple copies of that gene.
DNA sequencing. When a segment of DNA has been cloned, we can determine its nucleotide sequence. Nucleotide sequence is very important as it is the blueprint that contains the instructions for building an organism. Genetic function or evolution will never be complete without obtaining this information. As we already know, the recombinant DNA technology has led to powerful diagnostic procedures. useful in both medicine and forensics. In medicine, these diagnostic procedures are used in counseling prospective parents as to the likelihood of having a child with a particular disease. Researchers look for specific DNA fragments that are located in close proximity to the gene that causes the disease of concern. Not only that, but these diagnostic procedures are also used in the prenatal prediction of genetic disease in the fetus. In forensic, DNA fragments are employed to produce what is called a DNA fingerprint, and it can be used to determine if blood or other body fluids left at the scene of a crime belongs to a suspect.